Microbial control refers to the deliberate reduction, inhibition, or elimination of microorganisms to prevent disease transmission, spoilage, and contamination. At the highest level, sterilization is defined as the complete destruction or removal of all living microorganisms, including resistant forms such as bacterial endospores. Now, heat sterilization is the most reliable and commonly tested sterilization method because it denatures proteins and disrupts membranes irreversibly. The agent used to accomplish sterilization is referred to as sterilant, while commercial sterilization is a limited heat process designed to reduce spoilage organisms rather than eliminate all life. Below sterilization is disinfection, which targets harmful microorganisms on inanimate objects, but does not reliably eliminate endospores. Disinfectants may be chemicals such as chlorine, physical such as boiling water, or radiation based or UV light. In contrast, antisepsis applies specifically to living tissue, using chemicals that reduce microbial load without damaging host cells. Antiseptics, therefore, must balance antimicrobial effectiveness with tissue safety. Public health applications rely heavily on sanitation, which reduces microbial populations to levels deemed safe for public health standards rather than eliminating them entirely. This is commonly achieved through high pressure washing or chemical disinfectants. Closely related concepts include degerming, the mechanical removal of microbes from skin, such as alcohol swabs, and sepsis, which refers to microbial contamination associated with decay and infection. Microbial control agents function by targeting critical cellular functions. These include alternations of cell membrane permeability, denaturation of proteins, and damage to nucleic acids.
Microbial growth. Sterilization. Heat sterilization. Sterilant. Commercial sterilization. Disinfection. Inanimate objects. Does not kill endospores. Antisepsis. Living tissue. Antiseptics. Degerming. Sanitization. Public health safety. Reduced microbial load. Cellular targets. Membrane permeability. Protein damage. Nucleic acid damage.